Thank you.
Laura?
Hi.
Okay, Laura Splan joins us from New York. She's an artist working at the intersection of science, technology, and culture. Her projects are research-driven, frequently deal with biotechnology. She's currently developing a collaborative work with a theoretical biophysicist. Okay, that's it for my introduction. I'll let you talk. I'm sure you have more interesting things to say than just reading your bio.
Thank you so much. I'm going to go ahead and share my screen. And, oh, okay. Mouse. There we go.
Yes, I see it.
All right. Just close some of these panels. All right. Well, thank you so much, Piero, for having me back to speak at Laser again. I'm happy to be able to share my work remotely, even though I'd rather be on the Stanford campus again. And I have very fond memories of riding my bike around campus and teaching courses there. So tonight I'll be presenting some of my conceptually based work that explores virtual representations of the biological world through sensory encounters using a wide range of media. I frequently look to the molecular world to explore entanglements of cultural and biological systems. And I often leverage the material artifacts of science to reveal poetic subjectivities of the seemingly mundane. And I attempt to destabilize and reframe our relationship with the everyday and even redefine what the everyday means. I created this lace SARS doily in response to the 2003 SARS outbreak as part of a body of work grounded in the intricacy of textiles and associations with comforts of domesticity. But in the speculative narrative of a lace virus, collective traumas born from disease are rematerialized in an heirloom artifact to be passed on from one generation to the next. My research-based studio practice often includes residencies in labs and collaborations with scientists that explore scientific representations and technological abstractions of biological forms. And I often repurpose tools and techniques of science to engage with them both creatively and critically while searching for new possibilities. And I use fiber materialities and gestures in both real and virtual spaces to examine how our humanity is interwoven with the biological world and the cultural constructions by which we engage with it. Constructions that are so often mediated by technology. With a sensibility informed by textiles, I began to unravel antibody proteins in molecular visualization software. And as the proteins were manipulated, the software rendered uncanny disturbances. These videos are unedited screen recordings of performed interactions where my mouse is hidden and the labor of the user and the software is collapsed by the seemingly autonomous moving form. This animation, created with a model of a ricin toxin bound to an alpaca nanobody, is a collaborative doodle of sorts between the software, my hand, and the molecular structures that are being disrupted. Alpacas and llamas have unique antibody structures called nanobodies that can be, quote, humanized for human drugs. And these animals are sometimes referred to as living factories and are yet another abstraction in an increasingly complex biotechnological apparatus that entangles both human and non-human species. While sheltering in place for the pandemic in 2020, I continued these animations by incorporating molecular structures related to the coronavirus. This piece, called Denatured, is a screen recorded unraveling of SARS-CoV-2, its receptor binding domain, in complex with a llama nanobody. And in another series of animations, I began using a morphing technique to animate the folding and unfolding of the coronavirus spike protein that I was unraveling by hand. The imploding movement echoed the sense of precarity that was rendered so visible during the pandemic. I also became fascinated with the names of the colors available in the software that included references to nature, evocative of idyllic representations of the natural world, such as blue skies, green forests, ripe fruit, and romantic flowers. They present additional layers of abstraction built into the interfaces of the technologies we use to engage with and manipulate the natural world. The animations in my Unraveling series attribute their titles to the software's palette in order to reconnect these reductive correlations of language and color that allude to these idealized notions of nature. This animation is titled Lime, Limon, Forest, and its animated parts are SARS-CoV-2 spike proteins that are morphed animations transitioning with their unraveled forms, resulting in mesmerizing kaleidoscopic meditation, a mesmerizing kaleidoscopic meditation on invisible entanglements between natural and constructed worlds. And this is Orange Oxygen Salmon. In addition to cataloging the names of colors in software used to do science, I also collect metaphors and language used to explain science, particularly language that emerged to explain the biology of the coronavirus, and finding numerous sources for inspiration in the media, including this New York Times article called Bad News Wrapped in Protein, that's populated with a motley crew of the usual suspects in predictably dramatic narratives like saboteurs, tricksters, magicians, and eventually the mysteries that lie at the end of the line with a repeating chain of adenine nucleotides in a series known in biology as a termination sequence. I couldn't get this echoing melancholy of the repeated adenine nucleotides in the mRNA sequence out of my head, so I invited scientists and biotech workers to play the nucleotide sequence on guitar in recorded Zoom meetings, in which I prompted participants to play A on guitar 33 times. A live acoustic guitar performance of the sequence by biochemist Joseph Rucker became the backdrop for a 2020 installation at BioBat Art Space in Brooklyn, New York. His slow and persistent plucking of notes is at once soothing and stressful, invisible yet present. Another performance by lab instrumentation engineer Frank Masaki had an entrancing effect as he riffed on the sequence with an array of pedal effects and loops on electric guitar. Frank's recording, inspired by the materiality of the devastating coronavirus, served as a soundscape for an installation at BioBat Art Space. This installation was open to visitors through timed viewings, and visitors often described it as both meditative and womb-like. In September of 2021, 20 months after the first COVID death in the United States, I created a series of data-driven animations using statistical data from the pandemic. These animations were created with data tracking COVID deaths in the United States over the first 20 months of the pandemic to disrupt the 20 amino acid residues along protein structures. Each animation morphs from its biological folded form or conformation, as it's known in biology, to its technologically distorted form. The resulting disruptions create tableaus that are sometimes spastic and sometimes sublime. The animations intertwine models of the coronavirus with both human and non-human structures, including antibodies and cell receptors from llamas, alpacas, cats, dogs, pangolins, bats, and humans, evoking our increasing interspecies entanglements in the biomedical landscape. From zoonotic diseases to transgenic vaccine development, our understanding of what it means to be human in the natural world is becoming increasingly hard to define. These pseudo data visualizations rematerialize unseen cultural and biological forces that are inextricably linked to the technologies we use to understand them. During the bioart residency where I started experimenting with these molecular animations, my conversations with scientists about the use of non-human species for the production of biological products led to receiving over 200 pounds of fiber shorn from the laboratory llamas who produce antibodies for human drugs, including COVID vaccines. I spin their wool into yarn for handmade textiles, sculptures, and also installations that examine the hidden systems and invisible labor of biotechnology. One of these projects dovetailed with my interrogation of the language that's used to explain science. This doorway in an exhibition at the Esther Klein Gallery in Philadelphia included appropriated text from the book Molecular and Cell Biology for Dummies that equates plasma membranes with, quote, international boundaries and casts proteins as customs officers. The antagonistic implications of this metaphor were especially heightened by the zero-tolerance immigration policies being implemented during the production of this work in 2018. Viewers were instructed to move gently through a membrane of strands of llama wool. And the text on the wall reads, "The plasma membrane is the barrier between the cell and its environment. You can think of the plasma membrane as an international boundary, like the border between two countries. The molecules that act like customs officers are proteins. Receptor proteins receive signals on the outside of the cell and relay the message to the inside of the cell." So here biology gets co-opted as some sort of scientific justification for the enforcement of arbitrary boundaries circumscribed by wars of the past and the artifacts of colonialism that endure in the present. Scientific texts are often fraught with antagonistic metaphors that are grounded in narratives of trickery. In this one, after the virus is attached, it may trick the cell into bringing it inside. Or competition, smaller molecules will be closer to the finish line. Surveillance, defensive proteins wander by to make sure everything is okay. And assimilation, chaperone proteins guide proteins to their proper locations within the cell. In order to read the text excerpt and enter the next gallery, visitors had to pass through the curtain of llama yarn, and the strands had orphan keys tied to their ends that were collected from locksmiths and hardware stores in my neighborhood. And the lock and key metaphor is commonly used to explain antibody interaction with a cellular membrane and is evocative of the same tropes of the permeation of secure boundaries. As viewers permeated the boundary of the doorway, the keys signaled to viewers already inside that someone was entering from the outside. The gallery behind the curtain invited visitors to sit on a bale of hay while watching the molecular animation made with the model of an alpaca nanobody that illuminated the room, along with a neon sculpture that read, "Sit around and wait." The sign functioned as an invitation, a command, and a resignation, but also as a mantra for moments of uncertainty and anticipation. In a corner of the room, a motorized sculpture rotated every 90 seconds, mirroring the time it takes for a cell to form holes in its membrane during a process called bacterial transformation, when foreign DNA can enter a cell. Every 90 seconds, the spinning motor would agitate a spiral of chiming keys on strands of hand-spun llama fiber. I have also been experimenting with dyeing the hand-spun llama yarn using genetically manipulated bacteria. And these experiments with bacterial transformation serve as a conceptual framework to evoke notions of the transgression of boundaries, hybridity, and engineered bodies, while examining institutional notions of membranes as they're defined and performed by language, image, and movement. And while doing these experiments at the Coalesce BioArt residency at the University of Buffalo, I had the opportunity to teach a bacterial transformation workshop. And my goals for the workshop were to make the science accessible and demystify laboratory protocols and tools, but to also think critically about the language used to explain science. And the workshop included a discussion examining these metaphors and meta-narratives in biology texts. And then together we brainstormed for new language and metaphors to explain the science of bacterial transformation. While transforming bacteria in molecular space and unraveling proteins in virtual space, I continue to spin the llama yarn in my studio. And while cleaning and carding the wool, I often find clumps of feces, which I collect for this series of networked laboratory mixers. The machines activate when Twitter hashtags associated with the culturally contested status of science are tweeted. When taking office, the Trump administration advised how to improve the chances of receiving research funding with the suggestion to avoid phrases like diversity, transgender, fetus, and evidence-based. As the devices intermittently check for the latest tweets, the mixer's movements rematerialize the socio-political complexities of language. Here, the mere mention of hashtagged words like evidence-based or science-based agitates tubes filled with the laboratory animal feces. Another networked sculpture includes faint text on a wall that invites viewers to come close to read, "Our distance allows our intimacy." The phrase refers to the complexities of existence in the biotechnological age, where understanding of our own bodies and the bodies of others is increasingly mediated by technology. The sculpture blows a breeze in the viewer's face as they read the text. The speed of the networked fan intermittently adjusts based on the wind conditions near a farm in rural Pennsylvania, This farm is actually the 600-acre biological laboratory that gave me the fiber from the shearing of the llamas in their vivarium. On the other side of the walls on which the lab mixers and the fan are installed, viewers can see the network technologies of the devices laid bare while sitting on a rug made from the laboratory llama fiber. The installation creates a space for contemplation of the presence and absence of bodies and the mutability of categories that delineate their status. The rug from that installation was recently installed within a tactile sound installation that re-envisions an elevator as a biological cell and its visitors as proteins as they're prompted to engage with a haptic textile sculpture while listening to a sonic tour of a biotech laboratory. It includes the rug made from the hand-spun laboratory animal fiber with tactile transducers installed underneath that are connected to the soundscape entitled Chaperone. The transducers beneath the rug vibrate with the bumps, clicks, and bangs of laboratory machines in the soundscape. And the exhibition renders the elevator as a space for contemplation of the unseen labor of both humans and non-humans within an often invisible system. And with all my work, I'm particularly interested in how tactility and materiality can function to create an embodied engagement with the world that carefully considers what lies beneath the surface. And as our intimate connections with other bodies become increasingly mediated by technology, perhaps these immersive and interactive encounters with science can function as a rehearsal for interaction with and comprehension of the sublime magnitude of existence in the biotechnological age, which we both create and inhabit. Thank you so much. And you can find more information about these projects and my upcoming exhibitions on my website and social media. And I'm happy to answer any questions.
Yeah, not only the breadth, but the depth of your research is really impressive. Certainly unusual for an artist. I particularly like how you can bring together the biomedical world, which we associate, ordinary people associate with hospitals, with doctors, with diseases, with death. And you bring it together with aesthetics, which is certainly not an easy thing to do. I have a simple question. I don't know if the audience has a question, but I have a question actually that is more general. You interact, you're a case where you interact with a lot of things. You interact with scientists, you interact with an audience. I assume you interact with galleries, museums, you interact with the audience. What do you miss most from the pre-COVID world of all these interactions?
I think I actually miss seeing art more myself, like being an audience member more. And I mean, now things are opening up, but I just had more of a habit of being out in the world and seeing more art. It's interesting how my collaborations have changed. So I did that artist residency in Philadelphia at this company called Integral Molecular as part of the Science Center's BioArt residency. And that was in person in 2018. It was a 3-month residency for 1 day a week. And then I had the exhibition at the Esther Klein Gallery in 2019. And I had continued to work with the PyMall software to make animations. And when the pandemic happened and everyone was sheltering in place in March, April of 2020, I actually reached out to the lab to see if they'd be interested in a remote collaboration. And that was a super productive collaboration. And we interacted in very different ways over Zoom than we had in the lab. It just felt like a lot more kind of casual and familiar and conversational than when we were standing in the lab and around their research instead of like Zooming from our homes. So there are a lot of things I miss, but I also feel like I gained a lot of new ways of collaborating with people that that are very valuable.
And in terms of exhibiting your work, do you still see in the future relying on the gallery, on the museum, or have you found new ways to do it online?
Well, definitely during the pandemic, I have been in some online exhibitions where I've shown video and still image work in virtual spaces through Mozilla Hubs and some other platforms. But I'm really interested in how the gallery can serve as some sort of familiar template to address ideas of virtuality. And I'm just starting to kind of research this more, but I think the galleries and museums are actually kind of virtual spaces in and of themselves. Like they have already functioned as a rehearsal for us about how to enter a space that has its own rules. And you're always kind of, having to learn how to navigate the space on whatever the terms the artist has laid out. So yeah, I actually think there's a lot of potential for galleries and museums to serve a role in exploring our relationship with virtual spaces in the metaverse.
Excellent. Thank you very much.